product evolution curve, I don't know, in whatever field. So this is just a snapshot of what really happens at that end. Alright, for those of you who are really interested in microbiology, you can go beyond looking at which bacteria are in mursik and what do they do, because my grandmother doesn't know it, but she still makes the best mursik anyway. So it might be that this is not an innovative area anymore, but at the moment we are looking at cells like this. So if you dry cells and you measure residual water over intact cell activity, you have a moment in those cells where the survival rates, the metabolism, and the membrane integrity. When the membrane integrity comes down, that means the membrane is injured, the stability, the metabolic activity of those starter cultures also drop. And when that happens, the survival is also dropped. So this is the interaction we look at. We look at how does membrane integrity of your bacterial starter culture and the metabolic activity of those starter cultures vis-a-vis the survival of those starter cultures relate, for example, to the drying process. And we do this using some scanning microscope. This is, for example, from Lactobacillus helveticus. And a colleague from Thailand, he just graduated. In Germany, we don't pronounce the H, so don't imagine I am shrugging. Okay, he's from Thailand, and he just published a paper a couple of years ago, so you can have a look. Now, so, is something happening? Yeah. So loss of viability of the starter culture is very clear during a critical water content range of approximately 0.3 gram water activity per gram dry weight. And the major inactivation site in the cell is the membrane. So that is the focus at the moment. What do we do with the integrity of the membrane to protect the cell? Do we reinforce it? Yes. How? Here. If you have drying, what is green here is done at 40 degrees for 10 millibar. The circles in red, some of them are visible, is 30 degrees, and what you see that is relatively stable is at 15 degrees. So if you have residual water measured over a length of the storage period here, and you measure the survival rates, the ones that are done at 40 degrees come down so fast, I mean they reduce so fast, the others are not. So the viability of Lactobacillus paracasei here was shown after vacuum drying. Now contrast what you see there with here. When you have a protectant, and here you have a sugar called sorbitol, which is 25% weight by weight. So it's quite simple, just mix your starter culture with sorbitol, mix them and dry it. So that drying in the presence of sorbitol, 40 degrees, you see it comes down here, it doesn't quite move. So this is quite clearly an indicator that sorbitol has a very remarkable protective effect during drying of starter cultures. And the reason is because they protect the integrity of the membrane. So these are some of the products that you can get from hydrolyzed foods. Now I'm moving to an area which I'm so passionate about. You can get all these products if you do hydrolysis. I was just giving you an idea of what goes on in different fields. So that was microencapsulation followed by starter culture drying. Now here we are on hydrolysis. You can make toothpaste with peptides from casein or cheese. And the reason is because they have calcium, which is linked to phosphorus. And that makes it physiologically soluble in your mouth. At that pH, normally it wouldn't be soluble. That solubility makes the calcium bioavailable. And the bioavailability makes your teeth as white as mine. I don't use it, but I drink lots of milk. So this is one of the products that are produced using casein phosphopeptides. Casein, yeah, these are from casein, they are phosphor, that means they have phosphorus, and they are peptides. And by definition, caseins are proteins in milk which have a unique property that they all have phosphorus. So the phosphorus in casein linked to casein, if you introduce a calcium ion, a divalent ion, you have a casein phosphopeptide which is reacted with calcium, and that makes the toothpaste bioavailable. Some of these are available in Europe but slightly expensive, so while you are sitting in the morning and brushing your teeth, the teeth is saying, okay, thank you, and they are grabbing the calcium for formation of some enamel which would otherwise be reduced. So this is where we have lots of potential, ladies and gentlemen, here in Africa. I think even if we don't consume, there are others who have a lot of idle money, we can make it and send it to them. Okay. Evolus is a manufacturer, so this is made in Portugal. These products here have amylpeptide from Calpis in Japan. This product is claimed to help you lower high blood pressure. So if you drink lots of it, it has a sequence of amino acids, isoleucine, proline, or valine, proline. They have this unique property that they have a dipeptide from proline, and the proline, as you might know, is not an amino acid, it is an amino acid. That means it has a kink. It's not really a normal bond. That kink makes it resistant or inhibitory to an enzyme. And that enzyme, which is a big problem to people with high blood pressure, I'm sorry that here there is no blackboard where I could write, but normally I like writing. So we have an enzyme in our body which is responsible for high blood pressure, and this enzyme is called angiotensin 1 converting enzyme. This angiotensin 1 converting enzyme is a critical problem to people who are prone to high blood pressure, because there is an amino acid which is not an amino acid but a hormone, angiotensin 1 converting enzyme. You can just write it in short as ACE. So it has, there is a peptide which is very good for regulating our blood pressure. ACE converts that peptide from an 11 amino acid to a 9 amino acid. So it cuts off 2 amino acids at the end.